Okay, so good afternoon, everyone. I hope you are seeing the splash screen that talks about today's lecture. So welcome to the Lunchtime Lecture Series from the Botanical Research Institute of Texas and the Fort Worth Botanic Garden. I'm Dr. Brooke Best, Director of Research Programs, and I and my research colleagues host this lecture series, which takes place the first Tuesday of each month. Our next talk will actually be on it'll be a Friday forum. It's an ad hoc talk, and it will be on Friday, August 27th at 4 p.m. when Brit Research Associate Dr. Lisa George will present her work on the effect on native bees of planting experimental wildflower refugia near small-scale farms. And then after that, we'll feature on September 7th, I believe, Dr. James Beck, who's also a research associate from Wichita State University. He and a student will be presenting their work on invasive species, I believe. We are recording this talk, and we will we'll make the recording available on our YouTube channel as soon as we can and post the link on the event page. And sorry, getting some alerts here. Viewers, we'll ask that you remain muted throughout the lecture, but you can type your questions here anytime. And today, actually, since we have two guest speakers, we are going to run those back to back, and then we'll do all the questions and answers at the end. Okay, so one last comment. If the connection's getting a little choppy, I ask that you first try to turn off your video, and then you can, if that doesn't work, you can exit out, leave the meeting, and then you can rejoin with the same link. Then you can see if that fixes the issue. Okay, so I would like to present today's guest speakers. Today this is part of our student series, and we have two graduate students, or former graduate students, from University of North Texas, just nearby, just up the road in Denton, Texas. They both are from the lab of Dr. James Kennedy, who is a real champion in the natural history collections circles because he's done so much for those collections there on UNT's campus. So, Dr. Kennedy, we salute you. Our first speaker out of the two today will be Katie Vasquez. So, Katie was an undergraduate at UNT and continued on. We're doing a master's program with Dr. Kennedy in the Aquatic Ecology Lab. I believe she will be done within the year and is looking for future opportunities. So if you have anything you'd like to contact her about, any upcoming opportunities, I encourage you to do so. And perhaps she'll share her contact information there at the end, or I believe it might be on the event page as well. So let's go ahead and get started. Let me stop my share. And Katie, if you can share yours.
How's that?
Looks great.
Awesome. Okay, good afternoon, everyone. Thanks for having me. My name's Katie Vasquez, like Brooke said. I'm a student at UNT in Dr. Kennedy's lab. I study aquatic ecology and entomology. And my project is a co-op with the City of Austin's Watershed Protection, the Corps of Engineers in North Texas. And my master's research is over the macroinvertebrates of Lady Bird Lake and Lake Austin, Texas. And this is going to be research based on post-Hydrilla innovation in these lakes. Okay, so a really fast roadmap of what I'll talk about today. I'm just going to give a brief background on some of the lake challenges that both lakes have faced. This is primarily referring to the invasive species, the restoration efforts that have been done in light of this, and then talk a little bit about the importance of macroinvertebrates and macrophytes, what they are, why we use them, and then how this all ties together with my research, my big questions, and some of my objectives. Okay, so here are just some Google Maps of my sampling sites. Lake Austin is upstream of Lady Bird Lake. So both of these are part of the Colorado River system. Up here is Lake Travis, just to kind of help orient yourself if you know anything about this area. There's the Mansfield Dams here. Basically, Lake Austin is a very sinuous, strange looking lake. You wouldn't really look at this and think it was a lake, but it is. and then it feeds downstream into Lady Bird Lake. And then these red rectangles represent my sampling sites, just to kind of show you that they do span across the upstream of Lake Austin all the way downstream to Lady Bird Lake. And what kind of makes this setting a little unique is that it is in Austin, Texas, which is one of the fastest growing cities in the last 10 years in the US. It's estimated 100 people move to Austin every day. And there's a lot of pressure from the surrounding community, these lakefront properties. A lot of the residents are pretty high dollar, celebrities or athletes, some of them being Matthew McConaughey, Sandra Bullock, Joe Rogan has the new $14 million mansion, waterside. So there's a lot of pressure from the surrounding communities to upkeep the management of these areas and, kind of stay on top of all of this. So Lake Austin and Lady Bird Lake are both heavily sought destination sites for both locals and for travelers. Recreation on this lake is the primary resource that people use them for, anywhere from canoeing to jet skiing, wakeboarding, and then one of the biggest economic revenues for these lakes is game fishing, and specifically for largemouth bass, Microterra salmonides. Lake Austin was once considered one of the top 10 destinations for game fishing for largemouth bass in the US. And so there's a big demand on upkeeping the environment and the community of these fish is going for the economic revenue that it generates. So this kind of goes segue into the big issue of why my project even started. So there is an invasion of Hydrilla verticulata in Lake Austin. Back in 1999, they noticed that this plant, which is native to Asia, moved into the lake. And what makes this plant so invasive is it's very prolifically it grows very prolifically via fragmentation. So if a small piece breaks off, that small piece grows into a larger piece, and so on and so forth. So removing it mechanically is basically impossible. And so this plant grows and it clogs up boat motors and it impedes with flood control, clogs up the dams. So they had to find a way to get rid of this plant. And they did that by introducing the triploid Asian grass carp. So the Asian grass carp is native to East Asia, and it is a great alternative to biocontrol methods such as chemical herbicides. So instead of using chemical herbicides, they use the grass carp, and it is sterile, so it can't reproduce, and they do this to the eggs via pressure or temperature chemical control. And there is a stocking rate of this fish, so they do an evaluation of the plant coverage, and 50% or more, it's 5 an acre, and then 5 less if it's 50% or less coverage. And what makes these guys so great at being a biocontrol agent is that they are sterile, meaning that they can allocate the resources that they use for reproduction into growing. And they can grow much larger and larger faster than non-sterile fish. And then they've been shown to grow anywhere from 80 to 100 pounds. And what allows them to grow so fast is that they have these pharyngeal gill teeth, which are these like rows of comb-like teeth that are on the gill arches in their throat. So they're just consuming vegetation, grinding it up in their throat, and then just eating all day long. And they are so good at what they do that they actually ended up overgrazing in Lake Austin. So they not only ate all of the hydrilla, but they ate all of the native vegetation as well. And then some issues started arising from the decrease in native vegetation. So you get a decrease in water clarity, which you get an increase in erosion. You start seeing species decline of those game fish like the largemouth bass, and then you start seeing increased rates of eutrophication. We also saw the influx of zebra mussels, another invasive species. This is a filter feeder that can filter about 1 liter of water a day. And these guys really mess things up. They impair recreation and other water systems by fouling boats, utility pipes, beaches. And here's a good example of that. This is a shopping cart that was pulled out of one of the lakes. It is absolutely just caked with zebra mussels. And so this is kind of a good example of how bad they can be. There's also an increase in cyanoharmful algae blooms, These not only create aesthetic problems and reduce water clarity, but they're odorous, they impact recreation, and they can cause fish kills by when they decay, they consume all the oxygen out of the water. They also are known to generate hepatotoxins and neurotoxins. Oscillatoria is one of the genus of harmful algae that was found in Lady Bird Lake, and it's known to create a toxin called anatoxin A, which is a neurotoxin, and if consumed, it can result in death. And that's actually a lot of dog deaths happened in 2019. So they had to basically ban swimming and pets in the water until that got under control. So that's another issue that's been arising. And so a way to combat all of these issues that arose from the invasive plants and then the overgrazing of plants is to restore those baseline native communities back to the lakes. And so the restoration efforts basically are, they revolve around building enclosures, protective enclosures for just new plants that were planted into the riparian zones of these lakes, and that will protect them against the herbivory of turtles and fish. Some of the species that were restored into the lakes include water willow, pickerel weed, pondweed, eelgrass, and switchgrass. And then after the restoration efforts, basically those restoration efforts were assessed with my project, which is based on the macroinvertebrates that are living and residing within those communities. And here's an example of one of the enclosure pens that we built. You can see there are just kind of like little dots of plants there. That's freshly planted aquatic vegetation. And you can see there's a lot of barren sandy substrate. And the hopes is when we plant within these pens is to get them to spread and make a nice carpet-like founder colony situation so they can help stabilize that substrate and just take off. Here's what those pens look like kind of from the canal or the center of the lake. And this is a really good example of, this looks like eelgrass or vallisneria that's established a really nice carpet there. And then this is a really successful pen of what looks like Illinois pondweed and eelgrass or some water willow in the back there as well. And then this is all the way downstream at Lady Bird Lake. This is one of the vegetation pens there where it's really nice, lush green, stable vegetation. And this is just a picture of seasonal differences, what some of the sites can look like. And then one of the really fun challenges we get to deal with in aquatic restoration, at least on Lake Austin, is there's this issue of erosion. So upstream in Lake Austin is a wake zone, and Lady Bird Lake downstream is a no wake zone. So there's a huge dynamic between the amount of site disturbance upstream than downstream. And it is known that flow regime and current velocity do have an influence on the establishment of macrophytes and their success. And this is just a little video, make sure the volume's down, of kind of what this looks like. So a boat had just gone by, a wake boat, and there's this huge wave action that happens and crashes down into the site that we're trying to restore the vegetation to. So, you know, you have the small scouring and dislodging effect, and then of not only the plants, but everything that's trying to inhabit them as well. So there's definitely an observable difference between the upstream sites where this high erosion happens and the downstream sites, which is much more like a calm, less erosion, stable riparian system. Switching gears, a little bit about the macroinvertebrates. This is what I study. And in the scope of this project, you can think about macroinvertebrates in a sense as the primary link between the plants and the fish. So a way to assess the success of the restoration efforts is to look at the communities that live within them. And essentially you can think of it again as if you build it, they will come. So we restored the plants and now we're looking at what lives in them. And what is a macroinvertebrate? So macro is just referring to anything you can really catch within a 500 micron mesh sieve. Most of these guys are benthic and are comprised of multiple phyla of arthropods, mollusks, crustacean, annelids. Why use them? They are ubiquitous. You can find macroinvertebrates worldwide. They are wonderful indicators of environmental quality because of their growth, reproduction, and morphology. They have a range of tolerance and intolerance to certain conditions. You can think of water chemistry as more of a snapshot in time, whereas biomonitoring is more of like a moving picture of what's going on in the ecosystem. And this is because of their long life cycles, and they occupy multiple functional feeding groups that allow those connections to happen between different trophic relations. And they are important. It is known that macroinvertebrates do contribute a significant proportion to the material cycling and energy flow in aquatic systems. A great example of this is the non-biting midge larvae, the chironomid. It is known that most aquatic predators do feed on chironomids at some point in their life cycle, and that the larvae, pupae, and adults do form an integral part of the food web and act as food for other fish, invertebrates, amphibians, and stuff like that. So very important. And then submerged aquatic vegetation is the other key player in my research. Aquatic vegetation and aquatic macrophytes play multiple roles in aquatic systems. They are involved in nutrient uptake and retention, substrate stability, water clarity. They help attenuate all that wave energy, and they also provide food niches and refugia for fishes and macroinvertebrates. And it is known that macrophytes do support a higher macroinvertebrate richness, diversity, and abundance, and that is essentially what I'm looking at in these restored areas in the lakes. And that segues into my research. So my big research question is essentially, are these aquatic macrophytes supporting the macroinvertebrate communities? And I'm looking at this question in relationship to reservoir comparison. I'm looking at it between different habitat regimes, where a habitat regime is just referring to no plants, bare, mixed assemblage of plants, or a monoculture of plants. Looking at this seasonally. I'm also looking at this in relationship to individual plant species or assemblages and plant architecture, the density of plants, and then site disturbance. So this is more of an upstream, downstream thing. And how I do this is the two primary collection methods, one being the throw trap, which is essentially, it's a PVC cube, so to speak, that's lined with ripstop fabric, held on with fabric clips, and this allows me to generate a density of macrophytes and macroinvertebrates within a very specified sampling area. So basically swiping a D-frame net and collecting the vegetation within this area, and then I can quantify that. Here's just another picture of what that looks like. And the other primary collection method is the Hester Dendy. This is an artificial substrate sampler, and this is wonderful because it's supplemental to my other sampling method, because this sits out in the water where you designate it for weeks at a time. So this allows the colonization of what's living in the area within those plants to occupy those niches within the sampler. And then you can essentially scrape off everything that's living in it and process that as a sample. And this is primarily to collect those chironomid larvae that I was talking about earlier that are so important. And of course, all of the chironomid larvae were mounted and identified. You have to clear and mount these guys on a slide to identify them with their mouth and face parts. And this is done to the genus level. And this really helps refine the taxonomic resolution of the macroinvertebrate community and provides a lot of information. And then essentially the sample processing of macroinvertebrates kind of looks something like this, where you each sample is picked through. It's very a time extensive type thing, but you get to pick out all of the different taxa, you ID them to the furthest taxonomic level you can, which is typically the genus level, and then you use that in your data for community analysis. And so that's kind of all I prepared today. I do have some data handy if anybody has any questions about anything I've talked about. So that's it.
Thank you, Katie. That's fantastic. I love a good ecological study that has a ton of parameters. Nice and complex. We love those. I'll say for those of you who are afraid that you might forget your questions, you can type your questions in now, and so you don't forget them, and we will come back to those later after Megan's done. Okay, so let's move on to our second talk. I love that ending picture, by the way. Our second talk today is from, let me get my notes. I know who you are. I'm just want to make sure I get it right. Okay, our second talk today is from freshly minted PhD Dr. Megann Harlow. Megan was a PhD student in environmental science up at UNT, and she did her bachelor's and her master's at Hardin-Simmons, also kind of down the road. We've had a collaboration with Hardin-Simmons working on their herbarium specimens. So congrats. She recently defended in just this past April, and is going to be, I believe, maybe already starting or starting very soon, a postdoc at LAERF, which for those of you who don't know is Louisville Aquatic Ecosystem Research Facility. I believe I got that right, LAERF, L-A-E-R-F, which I believe is also a joint facility with the Army Corps of Engineers and is there kind of on the same campus as LELA, the, what is it, Louisville Lake Environmental Learning Area, all the acronyms. If I've gotten anything, any part of that wrong, please correct me before you go ahead. But Megan, let's go ahead and get started with your talk.
All right. Any problems hearing me?
Nope, you're good.
All right, so like she said, my name is Megann Harlow. My dissertation research is dealing with a biomonitoring study out at DFW Airport, where we were relating land use with aquatic life use. So I'm going to start out with a little bit of an introduction to urban streams and get into my study area. I'll go into a little bit of background on my methods and why we're using a land use analysis as well. I'll go over the major patterns of my results, take a little brief dive into taxonomic sufficiency, and then wrap everything. So urbanization and our aquatic ecosystems are mainly impacted through those changes on the landscape, mainly attributed to impervious surfaces, which are things like concrete buildings that prevent rainwater from flowing down into them. So that's kind of the main factor with urbanization. We also have development within that riparian zone where we're building right next to these rivers. We're removing that riparian vegetation, and that is really impacting our aquatic ecosystems as well. And so that's going to contribute to changes in our flow regimes and contribute to pollution. And so these kind of symptoms have been collectively coined and referred to as the urban stream syndrome. And so what we see in these streams is this altered flow condition, kind of depicted here on the right. You have flow volume on the y-axis and time on the x. And so after a rain event, you'll have in an urban stream these higher volumes of water and earlier volumes of water flowing downstream quicker after a rain event compared to a rural stream where there's less impervious surfaces, more riparian vegetation. You might have that same amount of water but spread out over a long period of time. And so that peak flow, that higher velocity is going to alter your channel morphology and stability. It's going to transmit more nutrients and contaminants from that urban environment. And all of these kind of contribute to a reduced biotic richness or diversity, where we have more species that are tolerant of pollution or some type of low oxygen condition. So the Dallas-Fort Worth International Airport is the second largest airport in the country in terms of land area. It contains over 17,000 acres within its property boundaries. It is located in the center of the DFW Metroplex. So because of this, it's often blamed for water quality issues downstream just because they're the biggest entity in the watershed. When we look at the literature, there's really not much specific impacts of airports on water quality, it's mainly treated as urban land use. Most airports aren't of sufficient size to be kind of treated any differently. And so these urban impacts are going to be related to the amount of impervious cover in these areas and any kind of non-point sources of pollution that are on the property. One unique aspect of airplanes or airports is their use of de-icer fluids to maintain safety and protect their equipment and people they're taxiing. So they will use those during colder weather events. And if these make it to our waterways, they can have a high oxygen demand and have been shown to have some toxicity. There are many mitigation practices. DFW employs quite a bit, and I'm sure it's standard protocol for a lot of these airports that use a lot of this material to have prevention practices in place to keep that from entering our waterways. So the DFW Airport has other watershed protection programs in place. And so with all of that information, they partnered with the UNT Benthic Ecology Lab to conduct biomonitoring studies on their area water bodies. This was initially done in 2004 with follow-ups in 2005, 2008, and 2014. My study is dealing pretty exclusively with the 2014 data set, which I want to point out is also in the middle of one of our most extreme droughts in the area in the last 10 years or so. And so we'll see why that's important in a little while. So the overall goals of this project were to characterize the water quality and the biological conditions, what's living in these streams, what is the quality, the ecological integrity of these streams, set the stage for a long-term bioassessment program, and evaluate the effectiveness of their many watershed protection programs that the DFW Airport had in place. So here is my study area. Let's focus here on the left first. We're located in between Denton, Tarrant, and Dallas counties. Our reference sites are more rural in location on the north and southeast or southwest side of the region. We are located in the Cross Timbers and Blackland Prairies ecoregion. So we are talking about prairie streams here. Hopefully you guys driven over probably or gone to the airport at some point and been around this area. So this flow ultimately into the Trinity River. The main stem here in our study area is Big Bear Creek. And I've divided my study sites into two categories to kind of simplify things. These main Bear Creek sites are here along the main Bear Creek channel. And then our runway sites are draining airport property directly. They're going to be smaller in size and kind of exclusively draining airport property. And I'll provide this diagram a couple of times throughout my talk today to help you keep your bearings. Okay, so my methods. We use standard biomonitoring procedures following the TCEQ's Surface Water Quality Monitoring Manual. That includes a habitat quality, water quality assessments, as well as a collections of the Benthic macroinvertebrate community. And we use those benthic macroinvertebrates to calculate an aquatic life use score, which I'll kind of go over briefly towards the end. But that's what we're doing. So Katie kind of touched on this a bit before, but I thought I would reiterate. It's kind of our emphasis in our lab. So why are we biomonitoring? What are we doing here? So we know that living organisms reflect their environment. If they're living there, they're able to persist there. And so they're providing, if we know ecological information about that organism, then we can infer that about the environment. So that's how they provide this long-term information, because they've been living there throughout their lifespan. Versus a single chemical measurement is just that one, like Katie said, snapshot or instance in time. And so if we're comparing streams, like say to our reference sites, and we see large differences that can, not always, that can indicate impairment or some kind of water quality perturbation or something. So we can compare that, our study streams, to the reference condition, or we can compare that over time to itself. And then again, what are benthic macroinvertebrates? Benthic just means they live along the bottom of the stream, but they can be living in the macrophytes, like with Katie's study. Macro generally means visible without a microscope. We can see these organisms. They're small, but we can see them. Here's a dragonfly nymph, a mayfly nymph. And so these are the juvenile forms of these organisms. You may be more used to a dragonfly out flying terrestrially, but these are what they look like underwater for much of their life. And so invertebrate includes these insects, but also amphipods, crustaceans, worms, things like that, leeches, all are included in this assessment. So in addition to the biomonitoring practices and methods that we used, I also did a spatial land use analysis. And the goal of this was to identify any variables upstream of my study sites that could impair water quality, as well as quantify the amount of impervious surfaces in the watersheds. And so this is what the results of that land use analysis look like. Most of the study area is this mowed herbaceous, about 43%. And so around the airport, that's going to be maintained mowed grasses. But in some of the other areas, it could be low scrubby vegetation or pasture land, or something like that. Throughout the entire watershed, the total area imperviousness was about 34%. And so that includes any transportation, roads and runways, driveways, parking lots, as well as buildings. And here's my Big Bear Creek site, so you can kind of keep your bearings here. When we look at those big main Bear Creek sites individually, you can see that they are very similar in terms of the amount of imperviousness. This floating number here is a combination of the buildings and the transportation. And so they're very similar. When you get to our runway sites, that's kind of what these look like. I've clipped out each area upstream of those. You get a little bit higher percent imperviousness and a little bit more variability. And then when you look at our reference sites, we see that they're much reduced in the amount of imperviousness that they have. And so I was able to detect differences between these three groups. And then when we look at my flow conditions from our water quality assessments, we noticed similar groupings as well. Our main Bear Creek sites had flowing water. Remember, this is a drought year, and so the water levels were low everywhere. But our main Bear Creek sites had the highest of the set. And then our runway sites had very low flow at the time of sampling and even no flow at 3 of our study sites. And so that translates to our macroinvertebrate community as well. We see those similar groupings again. Just to explain my graphic here, each color represents a different taxa. And so this is just the taxa composition for the main most abundant taxa that we have found in each sample. And then here on the bottom is a similarity cluster diagram. The shorter the line on the y-axis, the more similar these sites are. So these are my two most similar sites, and my main Bear Creek sites all were the most similar to each other. And again, these had more consistent flowing conditions, similar land uses, and any differences within those could really be attributed to the substrates that we found there. When we move away from this core 4 group, we have B19 and HBC. Those are our lower flowing sites. They get more unique, and our reference sites kind of move away as well. And then the most different over here are our non-flowing sites. You can tell by just the change in the color pattern here. It's an entirely different community. These organisms are adapted to non-flowing water. They're going to be present in that habitat. And so we can tell that they've kind of experienced that no flow or low flow for a while, for at least for the summer. So that brings me to the next group here. So the Chironomid community really clarifies this grouping between flows a lot more clearly. So we got the same thing going on here, but I'm going to point out just one major group, and that is the Tanipodini taxa. These guys only show up in my low flow sites. These guys are dominant in my continuous flow sites, and Larsia is showing up in my no flow sites. And so this pattern right here really brings me to a big portion of my dissertation, and that is taxonomic sufficiency. Katie touched on this a little bit. It's kind of a running theme in our lab also. But so what this means is how low do we need to go in taxonomic resolution? Is taking all the insects to the family level, is that good enough? Do we need that genus or that species level identification in order to learn the most or understand the most about our study systems? And so we've established that living organisms reflect their environment. And so we know a lot of ecological information about organisms at this genus and species level. And so when we condense taxa up into family or order, we lose a lot of that information when we're just making generalizations. And this is especially true for the Chironomidae family. They are a diverse group of aquatic insects. There's many different kinds. And despite what is kind of understood or stereotyped about them, they have a wide range of pollution tolerances and even feeding habits. So we're used to seeing the bloodworms and the things that can live in low oxygen conditions, but there are those chironomids out there that can need better conditions and can be more intolerant of pollution. And then in our area, they are especially dominant in our semi-arid prairie streams of this region. We get a lot of these organisms. In this study alone, we had 49 different chironomid genera present in my study, at least 49. And then in all our flowing sites, we had a range from 20 to 54% of their abundance. So they were dominant in most of our sites. And so how does that relate to biomonitoring? Most protocols in their procedures, they state that all aquatic insect families should be identified to genus except for the family Chironomidae. And ultimately, these guys are overlooked because it takes more time, it takes more resources to make that genus level identification. Like Katie said, you have to clear them and mount them. And there's a little bit of a learning curve in making those identifications, just like when you're identifying aquatic insects. So my question is, how does this methodology influence our bioassessment conclusions? Do we gain any more information by taking them to the genus level? Can we detect differences between sites that are very similar in their urbanness if we leave them at family, if we leave them at Chironomidae, or if we take them to the genus level? And so I'm not going to dive too deep into this, but I wanted to show this kind of pictorially. So here on the right side, this is a community analysis when you ignore the chironomid taxa, you just leave them at family. And you'll notice all of these y-axis lines are shorter. Everything is depicted as more similar, okay? When you include the chironomid taxa, you take them to the genus level, these lines are elongated, so you can detect those differences between sites. Look, even our most dissimilar ones, the difference 50% versus 30% similarity. So that's one reason why we recommend and conclude that genus level identifications are going to provide the most power in discriminating between sites, especially in streams that have similar biological conditions, high proportions of the chironomid family like those found in our area. So in particular for bioassessments, we could develop a complementary set of methods and metrics that gives investigators the option. If they know how to do it and know that's an option, if they have the resources available, then they can use that to really understand their ecosystems. So overall conclusions for the whole bioassessment study are the community, the benthic macroinvertebrate community, did suggest a high aquatic life use for many of our sites, especially considering the urban setting they're in and their drought conditions. And this just speaks to the resiliency of these streams. Our lowest sites that scored low in the ALU, again, that ALU is the aquatic life use score that is generated based on the benthic macroinvertebrate community. The lowest of those were driven mainly by the lack of flow and the drought conditions that we had. Here's a picture of one of my lowest scoring sites, Grapevine Creek, kind of very much like a ditch. but it was had very low flow to no flow at the time of sampling. This is all just standing water. And then our highest flowing sites, our highest ALU sites had flowing water, diverse substrates, and that is my most downstream site, Bear Creek. This is one of my favorite sites. It was really pretty, had great substrates. So the benthic macroinvertebrates really like this riffle habitat, good wadeable stream. So this is one of my exceptional sites. Ultimately, big picture, flow is the main factor distinguishing these sites from one another. More continuous versus more intermittent or lack of flow, especially during drought years. And as droughts become more common in our area, this will probably have implications for our streams in this way. So I'd like to thank the DFW Airport. Tim Wintercheck got this program started. Tammy Walters kind of kept it going, and all the others at the Environmental Affairs Department. I appreciate them and this opportunity. Dr. Kennedy for his help throughout this whole process. Dr. Bruce Hunter, Sam Atkinson, Barney Venables, and Gary Dick as were my committee members. Like I said, we defended in April, so it's been a fun semester. All of my Benthic ecology lab mates and bugsters who've helped out with this project over the years. There's been a lot of bug picking and chironomid mounting, and I'm really appreciative of everyone's help. And then my family who likes to come and pick bugs with me. So now I am, like Brooke said, at LAIRF. I'm dealing with invasive species, invasive plants, and doing research on biocontrol. So this is a picture of a Brazilian pepper tree, and there's a establishment of this plant out in South Texas, and we're looking to figure out ways to best raise this thrip and use it to control this plant. And if you have any questions, want to get to me directly, there's my email address. Feel free. Thank you guys very much.